Hello, my name is Jessica Escobar, and today I will be presenting my research titled A Computational Approach to Investigate the Role of Zinc Finger Protein ZF30C in Epigenetic Repression of Drosophila engrel gene. And before I begin, I'd just like to thank SCINBRE for funding and supporting this research. So starting with the introduction, polycomb repressive elements, or PREs, are DNA elements necessary to act as binding sites for Drosophila melanogaster PCG proteins to regulate target genes. And so these PCG proteins that I mentioned are polycomb group proteins, which are conserved epigenetic regulators that serve to sustain that transcriptional repression of target genes. And so the PRE DNA elements act as recruiters for DNA binding proteins, which then bind to PRCs via protein-protein interaction. And PRCs are just polycomb repressive complexes. And so for this specific investigation, we focus on the engrel gene, which occurs in the posterior part of wing discs and is silenced in the anterior part of wing discs by PCG repression of histones. And we're focusing on engrel because its PREs contain PCG recruiting DNA elements whose binding partners are yet to be identified. So if we focus on figure 1 on the right of the screen, the diagram represents the upstream region of engreled, which contains two PREs. The expanded region here is a part of the engreled PRE2, and it's about 181 base pairs, and we are focusing on a small part of that PRE. And so when site A was mutated, the PRE was no longer functional. And when a DNA immunoprecipitation mass spectrometric analysis was done, the magnified region shown was used as a bait to identify proteins that bind to site A, and this is essentially how we found ZF30C and identified it as a candidate protein. So continuing with the methods, this research's methods were essentially twofold. First, we did an evolutionary analysis using BLAST to identify key residues in ZF30C that could be conserved across species and possibly be important for DNA or protein binding. And so we split the protein into residues 1 to 400, representing the N-terminus, and then 401 to 777, representing the C-terminus, because this is where NCBI revealed that the region with the most conservation lied, which was somewhere around residue 400. And so the parameters for all of this are shown on figure 2 to the right. And then next we did a molecular dynamic simulation, which essentially uses an artificially modeled system to monitor the change of atom location and see how and where ZF30C binds to DNA or other proteins. And to do this, we used iTASR to make a crystal structure of ZF30C, and then use this model to compare to other known proteins with similar structures. And when we did this, we found a protein titled 2KMK, which is a known transcriptional repressor. We took interest in this protein because PDB indicated that it binds to the DNA region AGT-GAT, which is only one base different than the binding region we deemed as critical in the immunoprecipitation mentioned earlier. And so the structure of 2KMK was used for the MD simulation by using its DNA and the protein structures with ZF30C structures from the iTASR modeling, and also inserting the sequences AGT-GAA into the DNA helix, because as we said before, when this region was mutated, binding no longer occurred. And just to go back to that, this is that region. So now on to the results. The evolutionary analysis revealed a minimal amount of analogs for the N-terminus. All of them belong to the Drosophila genus, and so this wasn't very critical for us. However, the C-terminus had almost 2,000 analogs from a variety of genera, meaning that it could be partly conserved and important in binding. However, the results for the MD simulation were pretty interesting because the simulation was run over 40 nanoseconds, and we were able to observe that 4 helices interacted with DNA, all of them being between residues 557 to 680. And so if you look at figure 3 on the right, we can see the model that was used, and this represents the simulation of ZF30C binding to AGT-GAA, as said before, which was the critical binding site in figure 1. And it's important to note here that the zinc ions that are present, that most of them lie in the C-terminus where all of the interactions were made. And so I can point these out. They're silver spheres. I think I can draw. And they lie right here. There's one right here, and there's another right here. Okay. And additionally, for more results, I mentioned before that the MD simulation monitors the change of location of atoms in a solution in binding activity. And this can be measured using a root mean square fluctuation, which measures the position change of each residue. Root mean square fluctuation, we just call it RMSF. And so for an RMSF, the region with more fluctuation is where protein-protein interaction occurs, whereas the region that is more stable and has less fluctuation is where DNA binding most likely occurs. So in figure 4, if we look at this, we can see that the region with the least fluctuation and most stability is between residues 530 to 680, which indicates that this is likely where DNA interactions are occurring for ZF30C. And so in conclusion, the data from the MD simulation and the evolutionary analysis give us reason to believe that it is likely that ZF30C interacts with DNA around residues 530 to 680. And those two results really did align. And so in addition, a possible limitation that I'd like to address for this research is that the MD simulation is a computational method that works with smaller ligands. And this should be noted because ZF30C is a larger ligand containing 777 amino acids, as said before. And lastly, as far as future directions, the next step to continue our research computationally would be carrying out an analysis of ZF30C interacting with comb gap and other PRE DNA binding proteins that we know of. Thank you for watching.